Chapter 5, Section 2. Complementary interactions between proteins and ligands. The immune system and immunoglobulins. Antibodies. Yes, I know this stuff. Yes. So, we're going to be looking at the immune response. Okay, you got to remember the immune response is a set of interactions. It's a dance between different proteins, different molecules, and different cell types. Okay? The whole point of your immune system is to discern self from non-self. Anything self, it's not supposed to recognize, other than cells that say, hey, I am Chad. Hey, I am Chad's heart. Don't kill me. Anything not self, kill it. Kill it with fire. So this is how we eliminate viruses, bacteria, fungal, any pathogens, parasites that we come into contact with. You have what are referred to as the white blood cells, referred to as leukocytes. The leukocytes, the two primary ones that we focus on most is the macrophages. These are the cells that go and patrol throughout your body. Anything not you, they eat. Think Pac-Man, waka 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 waka. And then lymphocytes. Lymphocytes, they go through and they produce different types of cytokines and things like that that tell the rest of the cells, it's like sending up a flare. Danger, 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 infection, infection. That's their job. We see that there's two portions to our immune responses. These are the learned responses. First is humoral, okay? Humoral system. These are going to be directed at bacterial and viruses. Viruses that are outside of a cell host. These are the ones floating around in your bloodstream. They're floating around in your interstitial fluids. Then you're going to have the cellular immune response. These are going to be cells that have learned there's something there. They have learned that it is time to go find and kill something, not you. One of the chief things you have flowing in your bloodstream right now that is acting as a protectant is what is known as antibodies. Antibodies are proteins produced by the B lymphocytes, the B cells. Okay, they call them the B cells because they develop and begin their maturation in the bone marrow, B cells. The levels of these antibodies goes up and down depending upon how long it's been since you've been exposed to something, how often are these exposures repeated. We refer to antibodies also as immunoglobulins, okay? Globulins, not globins, like the hemoglobins from the previous section. The immunoglobulins, the globulin is a family of proteins that have relatively the same structure. And if you take for immunology, you'll see that these globulin structures are found throughout the body for many different things. Antibodies are produced by B cells, the B cells that learn something not you. We have to keep in mind, your immune system does not learn the virus, that does not learn a bacteria, does not learn a parasite. What it learns is part of a protein on the surface of the virus, the bacteria, the parasite. That's what your immune cells learn. That is what you make antibodies to. We also have the other side of that coin of cells, the learned response. These are the T lymphocytes, T thymus, okay? These develop, mature in the thymus. They start in the bone marrow, and then they travel through to the thymus where they learn things and they get processed, learn, self, non-self. Again, take immunology if you want to know more of those steps. So the T for T cells, thymus, they come in different flavors. What are known as cytotoxic T cells and helper T cells. Cytotoxic, cytocell toxic killer. These are assassins. These cytotoxic T cells, also referred to as CD8 T cells, when activated, will search your body for any cell that shows signs of being infected with a specific virus. Right now, the whole thing with the COVID and all that, the people they're focusing on antibody response, because if you got a good antibody response, they assume you're going to have a good T cell response. Antibodies are going to be binding all the free floating, COVID viruses in your bloodstream and your fluids. But those antibodies do not penetrate into cells, and they have nothing to do with the viruses in the cells. The cells that are viral infected will put up certain flags that tell your immune system, hey, I'm infected. They put out certain proteins. Here's proteins. It's not all. Some of these proteins aren't going to be self. Your immune system takes those different things, some of them are viruses, and they go to different T cells. Do you know this? Do you know this? Do you know this? If a T cell says, oh yeah, I've learned that, it'll go through and start looking for any cell in your body that it's showing that protein on its surface. T cell receptors interact with different receptors on the surface of cells called MHC receptors, major histocompatibility complexes. The major histocompatibility complexes take random proteins, cut them up, random proteins within the cell, cut them up, and display them out. The majority of the time that MHC is putting out self-proteins, your immune system, ignores it. Every so often, though, here's a virus protein, virus protein, virus protein. Cytotoxic T cells, the CD8 cells, come along. They're going cell by cell. Ooh, you've got something. You've got something I recognize. Time for you to die. They kill that cell. Kill the cell, it collapses in on itself, capturing any viruses that are inside it, will be cleared out by macrophages and other immune cells later. The helper T cells do exactly what the name says. These are CD4 Th T helper cells. The CD4 T helper cells tell the B cells, hey, something here you recognize, start making antibodies, and then they go and they also wake up and tell the cytotoxic CD8 cells, hey, there's something here you would recognize, hunt it down, kill it with fire. So the basis of the vaccines, and that's a big thing we're seeing nowadays, vaccines, is to introduce into the body, us, animals, whoever you're vaccinating, versions of the virus, the bacteria, the toxin, whatever, that have been rendered ineffective, uninfective, or weakened to a point that they can't complete their life cycles, and thus can't complete whatever damage they're going to do to you. It's introduced into your body, your immune system learns it. The initial time something gets introduced, whether it's a vaccine or a natural exposure, takes about 2 weeks, 10 to 14 days, depending upon your health, depending upon your state of your immune system. Have you seen anything similar to once before, whatever. So about 2 weeks that first time. But subsequent after that, every time you're exposed to that again, that virus, that bacteria, whatever, instead of taking 2 weeks to learn it, It takes less than 2 days for your body, your immune system to go, boom, 0 to 60 in less than 2 days. The first time you're exposed, you get a certain amount of antibodies, certain amount of CD8, CD4 cells active. The next couple of times, the next time, antibody levels are going to be 10 times higher. The number of CD8, CD4 cells is going to be much, much higher. And every time you're going to be maintaining that level. Boom, boom, boom. After it's cleared, everything drops back down to a resting level, still there. Still have antibodies in your blood. You still have some CD4, CD8 cells hanging out. Not as many. Next time you see it, boom, boom. That's the basis of vaccine. So that during that 2 week your body learns something, that's 2 weeks that pathogen can be doing damage to your body. Heck, you look at Spanish flu, COVID for some people, and many of these other virus infections that we now have vaccines or are working towards, really good vaccines towards. Exposed on Monday, dead and buried by Sunday. That's well within that 2 weeks that your body's trying to learn stuff. Same with bacterial infections. Okay? The bacteria, as soon as they're in, within hours of getting into your body, the infection's going. And it's going to take 2 weeks for your body to learn it, to be able to fight it off maximally with B cells, T cells, and all that. Something more than just macrophages. Yeah, there's going to be a lot of damage can be done. Many of your immune responses are based on what are referred to as antigens. Okay? An antigen, as we see here, is anything that elicits an immune response. Okay? It could be a molecule, part of a polypeptide, part of, or it could be the whole bacteria, the whole virus. It's just sending off so many signals and making so many different things happen that your body's just responding to it. Now, as I said in the previous slide, it's not that the whole bacteria, it's not that the whole virus is what your body is responding to. No. It's what is referred to as an epitope. Part of a protein, part of a structure, part of. Not the whole virus, not the whole bacteria, a small part of a bigger thing that's part of the much bigger cell, viruses, the bacteria, or whatnot. Immunoglobulins are a set of proteins that are produced by B cells in response to an epitope. Again, your B cells do not learn the bacteria, the virus, things like that. So you watch the news nowadays and they're talking COVID and they're talking antibody titers of COVID. People have got a high antibody titers, blah, blah, blah. It's not that the B cells learn the entire virus, even though it's ridiculously small and microscopic. No, they learn part of a protein on the surface of the virus. That's the epitope. That's what the B cells make a protein known as immunoglobulins, Ig, against. Turns out that there's 5 different types of immunoglobulin. The primary one you're going to find in your serum is what is known as immunoglobulin G, IgG. Big I, little g, big G. Okay? 4 polypeptide change. Too heavy, too light. They have different motifs, what they refer to as the FC, the constant domain, or motifs, domains, constant domain, and the FAB, which is the variable regions. You look to the left there on A. That Y structure is actually, that's a pretty good rendition of schematically what an immunoglobulin looks like. The blue chains, the 2 the motifs, two up on the arms of the Y, two at the base. All together they form domains. The motifs come together, form the domains. FC, FAB. The blues are, constant down there. The chain, the end of the arms, that's where you're going to see the variable regions. The FC portion, all immunoglobulins in your body have that same FC. The different immunoglobulins produced by different B cells, which are responding to different epitopes. The differences you're going to see at the arms, at the FAB portion. Turns out at the end of the arms of the FAB portion, that's where you're going to have the binding sites. We draw it as you see in A because, well, humans are stupid animals. But in reality, it looks more like B. B shows you the ribbon model. Alpha helices and the beta strands and all of that. The FC portion has absolutely no interaction with the epitope. The FC portion, the base of the Y, is there for structure, to maintain the shape. It's at the end of the FAB portions, as it shows there, you have the antigen binding sites. Remember the antigen, the epitope, tomato, tomato, as far as you're concerned. Yes, there are differences, but if you really want to get to the differences, take me for immunology. So you see, it's the ends of the Y is where the antigen is going to be bound. Now here, it's a free-floating antigen. That antigen could be part of a much larger protein on the surface of a bacteria, on the surface of a virus, whatever. The 5 different classes, A, D, E, G, and M. You'll see them sometimes designated by the Greek lettering, alpha, delta, epsilon, gamma, mu. My mind, I'm always IgA, IgD, IgE, IgM, IgG. Why do they sometimes designate between Greek and the IgA? If you're discussing between different mammals, you're discussing homologs, paralogs, analogs between different mammals, us and mice, us and goat, stuff like that. Now, what's interesting is it doesn't matter which one you're talking about, D, E, M, A, G, whatever, they're all based on that Y. I mean, look at the figure here. This is what IgM looks like, an artist's rendition of it. You take one of those, or you take 5 of those IgGs, and you link them together. You have what's referred to as the J chain, and then binding between the different FC portions. Difference between D, E, and A, foreshortened FC domains. Or, as you see with IgA, the FC domains of 2 IgGs, or 2 immunoglobulins similar in shape to IgG, are joined FC to FC. This way, not the pentamer ring as you see with IgM. So what, you say, what's the big deal with this? Well, turns out that, remember, you think back to section 2 when we were discussing the dissociation constant, and we were showing, okay, here's to the far right on our sliding scale was enzyme substrate. Further to the left at a much greater 10 to 1,000 times greater affinity was antigen antibody. 10,000 or so higher than that was the, biotin-avidin interaction. So antibodies are highly specific and have a relatively high affinity for epitopes, things not you. What we see happens is you see the virus, the red. Now, the antibody and the virus size have been altered. It's not realistic. but it's just to show, to make it easier for the artist to draw for our minds to see. The antibodies are singular proteins. The virus is going to be, 10, maybe 100 times bigger. The virus is also going to be about 100 to 1,000 times smaller than the macrophage. Okay, so relative sizes here have been distorted for aid in us seeing this. Virus gets in your body. It's a virus you've seen before, so your B cells are there. T helper cells, the CD4 cells tell the B cells, wake up, start overproducing again. You get the antibodies flowing through your bloodstream, through your interstitial fluid. Antibodies come into contact with the virus. They will coat the virus. By coating the virus can't interact with its target cell. Like, stupid analogy, but it fits. Come winter, you're going to put on big, thick gloves, keep your hands warm. Well, imagine trying to type on your phone, text with those big gloves on. It's not going to work because you can't touch the screen, can't get contact with the screen. Same thing here with virus coated, they call it opsonized, virus opsonized by antibodies. Can't touch its target cell, can't get in, no infection. But now you also need to get rid of it. Turns out, as they're showing here, the macrophages, remember Pac-Man, waka 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 waka, that eats anything not you. Well, it turns out they have special receptors on the surface of the macrophage. These receptors only bind Fc portions of antibodies. It makes it easier for the macrophage to waka 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 the virus. Kind of cool. Now it turns out that, as you see here, there is a slight conformational change. The variable portion, the end of the FAB fragment of our immunoglobulins, extremely high affinity, low KD value. And you see that there is a pocket within that variable region at the end of the Ys where only a certain amino acid sequence, a certain structure can fit into it. Has to be able to interact with the R groups. Interacts with the R groups, you get the induced fit even at the ends of the Y, the FAB portions, which makes the binding even more tight. This isn't, for all intents and purposes, don't think of this as reversible binding like you see with the enzyme substrate that we talked about, or the protein, hemoglobin, oxygen we talked about previous section. We want this to be as close to irreversible as we can without having to make covalent bonds. Because you want those antibodies to bind and stick. Now, it turns out antibodies are not just antibodies and not just antibodies. Yes, there's the 5 different classes, but what's the source? What's the source of the antibodies? As an immunologist, we discuss things polyclonal, monoclonal. Polyclonal means different B cells of different lineages learn slightly different varied portions or epitopes of that overall protein on the surface of the virus. So this is going to be a mix of different antibodies from different lineages of B cells. Why is that important? Well, each of the lineage is going to get a different portion of that protein and is going to recognize that portion of the protein as an epitope. Some of the epitopes are going to lead to greater affinity binding of antibodies. So you may get 5 different B cells from 5 different lineages, all against, say, protein on the surface of COVID, the virus that causes COVID, SARS-CoV-19. This one, roughly the same area, so overlapping of epitopes, but it's, it's a good affinity, but not great. This one, different areas, better, but still not perfect. This one, very low affinity, but still binds, but just not really good. That's the problem with polyclonal. What we like to do is we like to study things from an immunologist standpoint. We like to study things analytically from what are known as monoclonal. The initial B cell that learned the epitope. Once it's told by its CD4 cell, hey, wake up, your thing's back, it goes through what's known as clonal expansion. 1 becomes 2, 4, 8, mitosis. And they all start making the antibodies. All of those came from the original one, so monoclonal. And it's more homogeneous. They're going for the same epitope, same affinity. In the real world, in your body, is polyclonal better than monoclonal? Monoclonal better than polyclonal? No. There's no... Personally, I see no difference, in an immune response. Honestly, if it's something that, could be bad, I'd rather have the polyclonal with a bunch of them, that may not be the best and greatest of affinity, and it grunts all of them. But hey, you know what? Throw everything in the kitchen sink, add a pathogen before it kills me. Monoclonal is great, it's perfect in the lab. Speaking of, in the lab, we can use this affinity, this specificity for a part of a protein on the surface and blah, 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 as a means of discerning the presence or absence of different proteins in a sample. It's called Western blots. You go through, you run a gel electrophoresis to separate all the proteins in a lysate. Remember, lysate is, cells that have been lysed, and this is the collection of all the proteins there. Separate out by size. Well, you don't know, not only in that gel, where your protein of interest is, how much of it's there, has there been any modifications to it. So you transfer all that protein from the gel to nitrocellulose paper as a substrate, a background for you to do tests on. Then you do, as they're showing here, you use antibodies. One antibody looks for your specific protein of interest, that epitope inside the entire protein. Then you come through with another second antibody to the first. First one can be from mouse, as they're showing here, the red mouse against part of a protein that's part of a lysate. The green, well, that's a secondary. Goat that has been injected with mouse antibodies. So now goat recognizes the immunoglobulin as mouse as foreign, starts making antibodies against its FC portion. Sweet. You can go through and manipulate this and do different things. You see in B, that's what you're resulting. The gel to the left there, the SDS gel, they used, some sort of a protein stain, so you just see this mix of stuff. To the right, the immunoblot. Yes, there's a mix of different things, different lysates, different preparations, lanes 1, 2, 3. But you look in the immunoblot, the specificity for your protein of interest is such that none of the other stuff in lane 1, which is also now in lane 4, is present. None of the other stuff in lane 2 is present in 5. Specificity. You can measure presence, absence, and get a relative idea of concentration based on this type of assay.